Hello, my name is Todd Stueckle, and I'm a research biologist at the National Institute for Occupational Safety and Health, also known as NIOSH. I'm also the co-coordinator and co-chair of the NIOSH Chronic Disease Program. Today, I want to give an overview of the NIOSH strategy for hazard characterization of carcinogens. And as a disclaimer, the views presented in this presentation should not be construed as policy of NIOSH or the CDC. And the views presented here are my own. All right, so the National Institute for Occupational Safety and Health, also known as NIOSH, has a mandate to assure that every man and woman in the nation a safe and healthful working condition and to preserve our human resources. And its mission is to develop new knowledge in the field of occupational safety and health and to transform and transfer that knowledge into practice. It is part of the Centers for Disease Control and Prevention. NIOSH has a strategic plan, and this basically reports research and service goals to address a broad range of occupational health and safety standards. NIOSH has external advisory committees made up of subject matter experts to advise NIOSH on the type of research it should be doing. And we also have global collaborations, mainly for knowledge exchange, international committees, standard development, and training materials. The jurisdiction and research, NIOSH's jurisdiction is fairly limited. We are mainly a research entity and we supply recommendations. We are largely a non-regulatory agency, but there are a couple regulations NIOSH is charged with. NIOSH provides guidance and recommendations and are often used by other agencies or bodies to develop regulations. And so the research needs that NIOSH has are several, one being surveillance, exposure assessment, and epidemiology. Second is we conduct rapid screening and hazard identification, and we tend to use new alternative methods, NAMs for that. Next, we want to know about potency and point of departure, and we use NAMs and traditional animal models typically for that. And then occupational risk assessment, and also intervention research. And so if there's recommended intervention practices to reduce disease or injury, we actually conduct research to see if those interventions actually work. For carcinogens, this is a list of carcinogens, traditional ones on the left that occur in the US workplace, and it ranges from radiation UV light to engineered nanomaterials, polyaromatic hydrocarbons, metals, and the list goes on and on. But those are toxicants, but we also look at occupational activities such as welding, painting, firefighting, and shift work have been recognized as being potential risks for cancer. On the right-hand side, NIOSH is currently getting out of the traditional occupational safety and health box, and we're starting to look at how personal risk factors may drive risk for cancer, and also at the socioeconomic risk factors like income, healthcare access, and also location of workers, and in the relationship to resources to help either to reduce cancer risk. We're also looking more at working life continuum. Most workers are no longer in the same job for say 30, 40 years. Most workers now have a whole slew of jobs that they will have, and with each job come different types of exposures. We're also looking at social determinants of health and how that plays into carcinogenic risk. The NIOSH research agenda specifically for cancer are these four main sectors currently, including healthcare workers, manufacturing workers, mining workers, and public safety workers. If you look at some of the activities that we're doing under these, a lot of them look at how exposure levels contribute to cancer risk. So that's going out and measuring exposure levels, but also looking at how those known exposure levels may drive say tumor development in appropriate lab models. We also are interested in understanding how, say, work organization contributes to cancer. A little bit of background of how NIOSH has traditionally done carcinogenicity testing. We have traditionally used an in vivo model. This is usually a mouse 1-year bioassay using an initiation promotion model. And if needed, we'll use a rat 2-year bioassay. Starting about 10 to 15 years ago, NIOSH started an in vitro program using either normal human or mouse cells, and these cells are either primary or immortalized. And we tend to look at the toxicant of interest, identify key characteristics of carcinogens, identifying how much potential hazard is there. We'll use in vitro models to look at acute exposures. And then what we'll do is once we narrow down to a suspected carcinogen, we'll conduct subchronic long-term exposures in culture and then assess cell phenotype and behavior and see if they show signs of the hallmarks of cancer. For example, evasion of cell death, increased cell proliferation, invasion, migration, and angiogenesis. What NIOSH does primarily when we develop outputs and how that is disseminated and how we hope that is used. So our primary output is published peer-reviewed papers in the scientific literature, presentations, and reports. And we also, when needed, conduct risk assessment for recommended exposure limits. Secondly, we hope this raises short-term outcomes, and this is basically raising awareness in the scientific industry and also interested parties. Our main driver for putting out our publications is hoping that intermediate outcomes are developed by stakeholders. And so examples of this would be industries using NIOSH information to change their manufacturing process to reduce the risk of cancer. Another example would be that standards organizations will use NIOSH data to establish exposure limits or prevention strategies. And we hope that this whole process will basically reduce exposure to carcinogens in the workplace. An example of an intermediate outcome is IARC. This is the International Agency of Research on Carcinogens based in the World Health Organization. They have a monographs program that forms working groups of subject matter experts to evaluate carcinogenic hazards using human data, animal data, and also some mechanistic data, including in vitro NAMS studies. And IARC will evaluate potential carcinogens and label them as group 1, group 2, or group 3. And then just to show you how important NIOSH work is for IARC, on the right-hand side is kind of a rundown of the last 11 years of IARC evaluations. And you can see in the blue bars is a percentage of how NIOSH has contributed. So on average, we have 50% or more of the IARC evaluations actually are using NIOSH data. Our carcinogenicity hazard characterization process, first we ask, is there a high burden and need? And that kind of determines whether we will be doing more work down the line. If so, then we try to understand exposure levels, the epidemiology, we look at physical chemical characterization, and then we'll use a tiered testing framework. It's really hard to look at every single chemical, say within a class of chemicals. So we'll pick a couple of representatives, do some either in silico, in vitro, ex vivo work to do hazard characterization on that using the key characteristics of carcinogens and even hallmarks of cancer. Once we identify some potential candidates, and if we need to, we'll go into a mouse model and run an initiation and promotion study. Through both the in vitro and in vivo studies, we'll look at mechanisms. When needed, we will develop risk assessments. And at least for carcinogens, instead of a recommended exposure limit, what we'll do is publish a risk management limit for that carcinogen. An example of how this has been done recently with, say, engineered nanomaterials. If you know anything about engineered nanomaterials, there's about hundreds of thousands developed every year. And so a classic hazard characterization alone for cancer is not possible. Conducting a traditional animal model is highly costly. And so using alternative testing strategies or also NAMS, we tend to use in vitro quite a bit, is that we can rapidly expedite data, reduce costs, and also regulatory and address regulatory agency directives. So how we do this is we'll use primarily that in vitro subchronic transformation model. And so on the left-hand side of the diagram, we've matched up in vitro cell transformation in the process say tumor development or neoplastic development in vitro and how that aligns with the in vivo. And as you can see, we can initiate DNA damage in both models. We can observe uncontrolled cell growth or hyperplasia in both models. We can evaluate evasion out apoptosis. We can observe the formation of tumors or colonies in the in vitro model, and then also assess more cancer-like behavior, such as metastasis in both models. We feel that the in vitro models that we use can serve as a tier 1 like screening platform for neoplastic transformation and actually potentially be predictive. We've tried to shy away a little bit from the She and the BALB3T3 cells, mainly because they're a mouse model, but we do comparisons of those mouse or murine cell models to the human cell model. And we're always trying to assess whether our some subchronic exposure in vitro is actually predictive of in vivo, but also the human condition. Again, for our NAMS models, we rely on the key characteristics of carcinogens and hallmarks. And here I just have a list of the endpoints we try to measure. And through all these measurements, we try to inform development and adverse outcome pathways. But again, there's with every model system, there's issues with that. And so we're continuously trying to develop new methods. Right now, we're trying to improve our understanding of epigenetics in both the in vitro and in vivo systems. And especially in vitro, we're trying to find new ways of modeling the tumor microenvironment and the development of that tumor microenvironment that is really critical for tumor formation. And so when this is a kind of an overview of the principle of our in vitro to in vivo comparison and our paradigm approach. So with the in vivo model, we can observe disease development, distribution, deposition, and clearance of, say, particles or toxicants. And this is relevant to a human, in our case, we deal a lot with the lung, so the human pulmonary exposure risk. On the in vitro side, we can rapidly screen, we can do comparative assessments, do dose response, and we feel that in some cases it is predictive of the in vivo response. We can look at specific cell types or in vivo that can be difficult at times, and we can identify detailed mechanisms. And so if we compare across in vivo and in vitro models, we hope to fill knowledge gaps and inform workplace exposure risk assessment and prevention strategies. An example of how we've done this, we conducted a study with carbon nanotubes. On the left-hand side, we had 6 treatment groups. 2 of these were carbon nanotubes, so single-walled carbon nanotube, multi-walled carbon nanotube, and we used several controls. So we use asbestos as a positive control. We use ultrafine carbon black as a negative control, and with some other controls, we exposed for about 5 to 6 months in culture, evaluated the cells for some of the hallmarks of cancer, and these carbon nanotubes did exhibit some of the key characteristics of carcinogens. And one of the unique ways that we've attempted, and it seems to work fairly well, is to do whole genome expression signatures, really trying to understand the underlying mechanisms for that. So across the bottom there, we can look at gene signaling networks and cell function analyses. But the other thing we can do is try to identify a very specific slew of genetic expression markers for, say, certain types of exposure and how that relates to potential neoplastic transformation in culture. And through some comparative marker selection and class neighbor analyses, we cross-validate that. And looking at both the mechanistic evidence in gene signaling, but also some of the markers that are pulled out of the biomarker type of assessment, we hope we are developing robust markers for early tumor detection, at least in the cell type, how that may relate to the mouse. That seems like there is some correlation to mouse expression, tumors in the mouse. And then we're hoping that that is informative for humans in the future. Just to show that our in vitro does align with what we're seeing in vivo. So this is all in vivo data. Carbon nanotubes, as we all know, can interfere with mitosis. So that's on the left-hand side, is we have damage to the mitotic spindle, and you basically have chromosomes going different directions causing adverse issues with proper chromosome formation in mitotic cells. So we have mitotic spindle disruption that can cause neoplastic transformation and lead to hyperplasia and even tumors in vivo. On the right-hand side, a study that was just published about a month or two ago, looking at the classic multi-wall carbon nanotube, the Mitsui 7 carbon nanotube, looking at dose response. We can see a nice clean dose response in the red bars. Using the initiation promotion model, we see a clear hyperplasia in the lung, and that develops into an invasive adenocarcinoma, and it's a very clear dose response on the right-hand side of your screen. Right now, one of the studies I'm involved in is looking at firefighting foams. This is really a PFAS-driven type of project. Not just looking at PFAS-enabled foams, which are the aqueous AFFF foams, but also looking at the replacement foams that are PFAS-free. We've done some exposures to human kidney cells in culture, done RNA-seq to look at gene expression and transcript expression, and we've used the carbon 8-chain PFOS as a control in this study. And as you can see, there's some similarities in some of the diseases that are identified in our analyses, some very common but also key differences in cellular functions, canonical pathways, and also genes. So we feel that this is a viable way forward of using in vitro and really looking at mechanistic and seeing how that may align with samples that we have from firefighters. So just as an overview of where we are right now, we feel that in vitro models at NIOSH, the benefits are we can do this in a controlled environment and very rapid and repeatedly. We can look at different types of toxicants and a tiered testing strategy. We can do dose response and make those across model comparisons. The challenges are quite a few. We're trying to get a better handle on toxicokinetics and fate, trying to scale up to multicellular responses. We still don't have an intact immune system, which is really important for tumor surveillance and also the microenvironment. Trying to conduct better long-term exposures is always a challenge. And looking at that disease development time course, and this really speaks to our inability right now to do, say, microenvironment for the tumor formation. But we are dabbling in some of these areas, and so we have some work going on right now with air-liquid interface cultures. We've done quite a few studies with tumor spheres, cancer tumor spheres, using cancer stem cells and seeing how those cancer stem cells lead to tumor spheres and how that may contribute to tumor development. And we also have a couple studies done looking at some of the supporting cells in the tumor microenvironment, including fibroblasts. But we're very much interested in looking at lung on a chip, organotypic models, and also 3D lung sections in the future. So that's all I have for you today. If that's my contact information, if you'd like to reach out, if you want to know more about the NIOSH Chronic Disease Program and also our external council, and that's the Nordic Chronic Disease Council, please feel free to use those QR codes and find out some more information. And thank you for your attention and have a good day.